Hi, my name is Blair Hoding. I am a third-year biology major, and I will be presenting my research on a mutant fibrinogen that is unable to form fibrin that can improve kidney function in mice with sickle cell anemia. Sickle cell anemia is caused by a mutation in the beta hemoglobin gene. This is the main protein that makes up red blood cells, which carries oxygen throughout the body. This mutation causes red blood cells to change from a donut shape to a sickle shape. Red blood cells can then burst, causing anemia or lack of healthy blood cells. Other complications include repeated vascular occlusion episodes, inflammation, and lifelong complications. The coagulation cascade is a part of the pathway that occurs when your blood needs to clot, like when you get a cut. This process is an overdrive in sickle cell patients and occurs without any vascular triggers. Fibrin is the last step in this pathway, forming a mesh that can trap platelets to clot blood. This can be very damaging to sickle cell patients, as fibrin is insoluble and can accumulate in the organs. This thought led us to investigate that if a mutation in fibrin could reduce accumulation, which could lead to improved outcomes in sickle cell patients. In the normal coagulation cascade, which will be referred to as wild type, fibrinogen, a precursor, is converted to fibrin by an enzyme known as thrombin. It cleaves the soluble fibrinogen to form insoluble fibrin, which then accumulates and damages organs. A mouse line was developed with a mutation in fibrinogen, known as fibrinogen AEK. This changes the structure of fibrinogen so that thrombin could no longer bind and cleave to fibrinogen, leaving it in its soluble form, which would be unable to deposit into organs, potentially leading to reduced damage. In order to assess organ damage, we took kidney samples of our mice. A normal non-diseased kidney is shown on the left. The box shows a microscopic look at the tissue, showing its functional structure like the glomerulus, the filtration system of the kidneys, fully intact and working. The diseased kidney, shown on the right, is smaller, shriveled, with lots of fibrous scar tissue. The glomerulus and other functional structures are damaged. This led to our hypothesis that elimination of the thrombin cleavage site of fibrinogen will prevent fibrin accumulation, leading to improved organ function and reduced pathology in sickle cell anemia mice. To test our hypothesis, we needed to test this mutant fibrinogen gene in mice that have sickle cell anemia. This can be accomplished with a bone marrow transplant. Blood cells are produced by stem cells in the bone marrow, so replacing normal cells with cells that have the sickle gene will cause the mice to present as if they had sickle cell anemia within 3 months of the transplant. Mice were divided into two groups. One group contained mice that would receive bone marrow, where the donor mouse had sickle cell bred into it. The other group had mice that received bone marrow from a normal mouse to serve as control that will not develop sickle cell anemia. Each group consisted of mice with the wild type fibrinogen and some mice with the mutant fibrinogen. We will mostly focus on the mice that received the sickle cell bone marrow transplant and look to see if the mice with the mutant fibrinogen, highlighted in red, will have improved outcomes. We followed this mice for 1 year and performed kidney function and tissue analysis. Examining protein in the urine, known as proteinuria, is a great way to assess kidney function. The less protein in the urine, the better the kidney is functioning. Both control mouse groups contain low levels of protein in the urine. However, wild-type sickle mice had remarkably high levels of protein in the urine, whereas the mutant sickle mice had significantly low levels of the urine and were comparable to the control group, with a p-value of less than 0.01. This indicates that the mutant fibrinogen significantly reduces kidney damage. Kidney histology was also analyzed to look for physical differences in the kidney structure between wild-type sickle mice and mutant sickle mice. In these slides, we can see that a large area of fibrosis in wild type, marked with blue, is present. There are minimal blue areas in the mutant mice, indicating less fibrosis. In these sets of slides, we are examining the amount of ischemia, or lack of blood flow in the tissue. The wild type sickle mice have noticeably less blood flow, as shown by the overall paler color. The mutant kidney shows a much brighter color, indicating the area is receiving more blood flow. Lastly, in these set of slides, the glomeruli, the filtration system of the kidney, are outlined in white dashed circles. Red dots represent the presence of podocytes, a type of kidney cell. There is an increase in this number of podocytes, indicating kidney cell survival in these mutant mice. All of these differences are significantly significant, as shown by the graphs, indicating an overall improvement in kidney cell in the mutant mice. Our study demonstrates that fibrinogen AEK mutation improves kidney fibrosis, ischemia, and protects podocytes from injury or death, resulting in reduced proteinuria in sickle cell anemia mice. Thank you to listening to this presentation.